In the last lecture, we have seen the contribution of various scientists associated with the 19th century. However, in this lecture, we are going to see the contribution of scientists of the 20th century. The first one is Emil Fischer. He has been awarded a Nobel Prize for his pioneering studies that have been conducted in the proteins. The details of the studies are all we will see in the further classes. The next one is Edmund Buchner, whose contribution is again associated with the discovery of enzymes. How the enzymes have been playing a major role are present there in the living system, for which again he has been awarded a Nobel Prize. Most of the scientists listed here are all Nobel Prize laureates. Okay, their contributions have been properly honored through Nobel Prize. The next one is a Hill and Meyerhoff. They are the one who have found the type of metabolism that have been taking place there in the muscle tissue. That is, they are the first to identify the relationship between the muscle metabolism and lactic acid production that is happening due to the insufficient presence of oxygen there in the muscle tissues. So for this, they have been awarded a Nobel Prize. Then Swedberg. Swedberg is shortly we used to call this as S. If you look at into the ribosomes, it is commonly referred as a 70S ribosome and 80S. In that S stands for Swedberg's coefficient. Okay. So he is a person who has studied on colloid and developed the analytical ultracentrifugation. So the analytical ultracentrifugation process was first developed by Swedberg, for which he has been awarded a Nobel Prize. Next one is Landsteiner, who has been awarded a Nobel Prize for the discovery of the human blood groups, the importance of blood groups there in the human system. Knoll and Ruska, they are all basically Canadian scientists who have been associated with the building of a transmission electron microscope for the first time in the world. They have built a transmission electron microscope which is having a lot of application in the future studies. Then comes the contribution of Lewis. Lewis is a person who has discovered the pinocytosis. What is a pinocytosis? In a simpler way we can able to understand about the pinocytosis. You look at this image which shows about the pinocytosis. So pinocytosis is taking up of fluids and solutes inside, especially fluid. Taking out of the fluids inside are referred as a pinocytosis. Phagocytosis, I am just recollecting what we have studied in the previous lecture. Taking certain large particles inside the cell. Say for example, a bacteria has been engulfed there by the cell with the help of the pseudopodia formation by the plasma membrane. The next contribution is by Zernick. Again, he was awarded a Nobel Prize for inventing the phase contrast microscope. Phase contrast microscope is having a lot of application there in looking at the interior details of the cells. The details of this we will be studying there in the fundamentals of microbiology paper. And the next one is the contribution of Danieli and Davison. Danieli and Davison are the persons who have proposed the protein-lipid-protein structure of the plasma membrane. That is, they are the persons to first propose some kind of a structure for the plasma membrane. How this protein-to-protein structure is being present? It is commonly referred as a sandwich model of the protein. So this is a sandwich, okay. Same way in which the protein lipid will be present there. How? You look at the point here related to Davison and Danieli model. It is a model proposed for the functioning of plasma membrane of the cell. It describes as a phospholipid bilayer that lies between two layers of globular protein. It is a trilaminar and lipoproteinous in nature. Okay. How this will be looking there in a image. So this is a sandwich model. It is looking like a bread sandwich there. So that is the reason it is referred as a sandwich model. Here you can able to see the phospholipid present interior. In the outside you can able to see the protein. Again a phospholipid has been present there in the lipid bilayer. Inside that thing you can able to see a protein. So this model was proposed in 1935 by Davison and Danieli. The next one is Hans Adolf Krebs. Hans Adolf Krebs is related with the discovery of the citric acid cycle. Citric acid cycle is the one which is very important in the respiration process. or in the cellular respiration. So Hans Adolf Krebs was a physician and a biochemist and a biologist. He was a pioneer in the study of cellular respiration. That is a biochemical process behind the living cells. That is how the organism is utilizing the glucose in the presence of oxygen and how it drives the whole process of the life. So these findings have been provided there by Hans Adolf Krebs. The next one is Edmund Beecher Wilson. He is commonly referred as America's first cellular biologist. He is a person who discovered the sex chromosomes and their arrangements there in the human cells. Next contribution is by Konstantin Merezhkovsky. He describes the theory of endosymbiosis. Endosymbiosis theory can be best explained in this diagram. You look at this diagram. Okay, this diagram explains how the cell and the various cellular components have been formed there. Okay, if you look at here, an ancestral prokaryote is there, which is having just a cell membrane. Okay, it's an infolding of the cell plasma membrane have been shown there, which later forms into some specific structures such as a endoplasmic reticulum, nuclear envelope and nucleus. These are all the things that have been formed during the course of the evolution. Cell with a nucleus and an endomembrane system has been present there. This cell is capable of engulfing some kind of a aerobic heterotrophic bacteria. Some aerobic heterotrophic bacteria have been engulfed there, which has later evolved into mitochondrion through endosymbiosis process. Okay, so this has been later evolved into the mitochondrion. In the same cell, when it has a chance to engulf another photosynthetic bacteria, particularly some kind of a cyanobacteria, when it is engulfed, it forms there into the chloroplast structure. That is, they are showing as a plastic. It forms into the chloroplast structure of the ancestral photosynthetic eukaryotic cell. That is, an ancestral photosynthetic eukaryotic cell, the photosynthesis process is resulted due to the endosymbiotic origin, which is facilitated by engulfing of a photosynthetic prokaryote. However, the respiration there in a ancestral heterotrophic eukaryotic organism is facilitated by engulfing and endosymbiotic origin of the mitochondrial structure. So this kind of a evolution of some cell structures that have been present inside the cell, mainly the mitochondria and chloroplast. Both these things are having an origin there only from the bacteria. That is, eukaryotic cells have engulfed that thing and it has stayed there and it has further get evolved for some specific functions or from some specialized functions such as the photosynthetic phosphorylation, that is the one carried out there in the chloroplast, and a normal aerobic respiration is the one which is carried out there by the mitochondria. The next one is Theodore Boveri. He has identified the centrosome and it is described its role in the cell division. That is, the importance of centrosome there in the cell division was first proposed by Theodore Boveri. The last contribution is related to Peter Mitchell. Peter Mitchell is again awarded a Nobel Prize for its important contribution of hemiosmotic mechanism of ATP synthesis. That is, ATP can be synthesized by different kinds of a mechanism, say by substrate level phosphorylation, by a hemiosmotic phosphorylation that is taking place there during the aerobic respiration. Okay, so this hemiosmotic based ATP synthesis was first denoted there by Peter Mitchell. Detail of these things we will see in the subsequent classes. Lastly, we are going to see a few scientists who are all recent scientists and their discoveries that has shaped the cell biology. First one is Leon Margulis. Leon Margulis is an American biologist, a lady biologist who have proposed the theory of origin of eukaryotic organelles. That is the one we have seen just before in the lecture, how a mitochondria and a chloroplast have been originated. So she has provided a lot of supporting evidences for the endosymbiotic theory. She is Leon Margulis and she has proposed that she is an American evolutionary theorist, biologist and a science author and a popular science popularizer. She has proposed the a primary modern proponent for the significance of endosymbiosis in evolution. So she is the one who has proposed the evidences for the endosymbiotic theory. Okay, she is no more now. Okay, she is died in the year 2011. The next scientist is Günter Blobel. He was also awarded a Nobel Prize for the discovery of the protein targeting in the cells. To which particular place the protein is getting targeted in a cell was founded by his discovery. So for that he has been awarded a Nobel Prize. And the next important discovery is of Peter Agre. Peter Agre is the one who has proposed the presence of porins, some specific channels that have been present in the cells through which water or other solute molecules will be moving. So such structures are referred as a porins. So you can look at into the porine structure there. So in a broader sense, it refers to a class of protein whose molecules can form into some kind of a channels through which the passage of small ions and molecules will be permitted there in the cellular membranes. How these porins will be present. So these are all the structures of the porins. So they are all channels, some located there in the gram-negative bacteria, in the outer membrane of the gram-negative bacteria. So like that, porins are playing a major role there in the living organisms. The next contribution is by Ira Mellon. He is a cell biologist who have discovered the importance of the endosome structures there in the cell. We can go into the details of these things in the subsequent classes. And then the next discovery is by Gibson and Craig Venter. It is in the somewhat recent in the year 2010. They are the persons who have created a synthetic cell and named that particular cell as a Mycoplasma laboratorium. It was actually a assembled cell. If possible, in the subsequent classes, we will go into the detail of this first synthesized cell, which is named as a Mycoplasma laboratorium.